Welcome to Learnpedia, the ultimate JEE and NEET prep tool currently being used by more than 20,000 aspirants. Let's see if you can answer this important question. If you think you got the answer, post it in the comment section below. To understand the concept behind this question, go ahead and watch the full video.
Nephron. Nephron is the basic structural and functional unit of kidney. In man, each kidney has 1.2 million nephrons. It is thin, long, nearly about 3 centimeters, twisted tubule extending from the cortical to medullary part. It has mainly 4 parts. First, Bowman's capsule, proximal convoluted tubule, loop of Henle, and distal convoluted tubule. So, looking at the diagram, this is the Bowman's capsule. This convoluted tubule, which is proximal to the Bowman's capsule, which is nearer to the Bowman's capsule, is known as proximal convoluted tubule. This, which is a loop, a hairpin bend like, this is the loop of Henle. This loop of Henle has 2 limbs, which is the descending limb and the ascending limb. This convoluted tubule, which is away from the Bowman's capsule, hence it is known as distal convoluted tubule. The distal convoluted tubule opens into the collecting tubule, and the collecting tubule further opens into the collecting duct. Many collecting ducts open in a common duct of opens outside, and they converge and form the common duct of Bellini, which converges at the renal pyramid. So these are the parts of nephron. So the distal convoluted tubule opens into collecting tubules. Collecting tubules open into collecting duct. These collecting ducts open into common duct of Bellini. These common duct of Bellini, they converge at the renal pyramid. There are 2 types of nephrons, cortical nephrons and juxtamedullary nephrons. Cortical nephrons are the nephrons which are in huge amount. 85% of the nephrons constitute cortical nephrons. These have short loop of Henle which extended little in medulla. Juxtamedullary nephrons constitute 15% of the total nephrons, and these nephrons have long loop of Henle. These have long loop of Henle extended deep into the medulla, and their Bowman's capsule lie near the junction of cortex and medulla. Beginning with the structure of nephron, the first part is the Bowman's capsule. The closed end of nephron is pushed on itself to form a cup-like structure called as Bowman's capsule. This Bowman capsule is made of 2-layered squamous epithelium known as glomerulus and together called as Malpighian body or renal corpuscle. The epithelium cells of the Bowman's capsule are called podocytes. These podocytes are arranged in an intricate manner so as to leave some minute spaces called as filtration slits or slit pores. Each nephron has its own blood supply. From the renal artery, an efferent arteriole enters the glomerulus. This forms a knot of capillaries inside the glomerulus capsule, and the blood leaves the glomerulus through the efferent arteriole, which latter joins into renal vein. This is a cup-shaped structure known as Bowman's capsule. It has an epithelium with some slit pores in between, which are known as filtration slits or slit pores. These are the podocytes. From the renal vein, an efferent vessel enters. This is known as efferent arteriole. This efferent arteriole further forms a network of capillaries, and the blood leaves this glomerular space through the efferent arteriole. Juxtaglomerular apparatus. This is a region of contact between efferent arteriole and distal convoluted tubule. It includes 3 types of cells in the cortical part. First, cells around the efferent arteriole called juxtaglomerular cells. Cells around distal convoluted tubules called as macula densa, and the cells situated in the angle between efferent and efferent arteriole called Lassus cells or Volkison. So the three cells constituting the juxtaglomerular apparatus is the juxtaglomerular cells, macula densa, and the Lassus cells or Volkison. Importance of juxtaglomerular apparatus. Collecting tubule. This is about 20 mm long, straight tubule. Many of these tubules join to form duct of Bellini. In this diagram, we can see a Malpighian body. This Malpighian body comprises of two structures. One, the cup-shaped structure called Bowman's capsule, and the meshwork of blood capillaries called the glomerulus. So the Malpighian body is made up of two parts. Bowman's capsule, which is cup-shaped structure, and the meshwork of capillaries called as glomerulus. Malpighian body comprises of cup-shaped capsule known as Bowman's capsule and a meshwork of capillaries known as glomerulus. This Bowman capsule further leads into the renal tubule, which becomes convoluted or coiled in the cortex. So this is the coiled tube, and this tube is named as proximal convoluted tubule. So Bowman's capsule further leads into PCT, that is proximal convoluted tubule. Then this goes downwards as the descending loop of Henle. This is the loop of Henle. This is the descending limb and this one is the ascending limb. The descending loop of Henle gets narrowed in the region of medulla as a thin segment and then turns upwards as ascending loop of Henle or ascending limb as a thin segment. This thin segment further thickens and proceeds towards the cortex and forms the coiled tubule known as distal convoluted tubule or DCT. This distal convoluted tubule opens into the collecting duct. This distal convoluted tubule opens into the collecting tubule. Many collecting tubules of a number of nephrons open into a bigger duct known as collecting duct. The collecting duct efferent nephrons further unite to form a still larger duct called as duct of Bellini. These duct of Bellini run through the renal pyramids and open into the renal pelvis. Coming to the differences of cortical nephrons and juxtamedullary nephrons. Cortical nephrons comprises 85 to 86% of the total nephrons. So cortical nephrons are more in number, whereas juxtamedullary nephron comprises 14 to 15% of the total nephrons. The glomeruli are smaller and located in the cortex in case of cortical nephrons, whereas in juxtamedullary, glomeruli are large and located at the junction of cortex and medulla. In cortical nephrons, the loop of Henle is short and penetrate less deep into the medulla before coming back into the cortex. So they are short and less deep, whereas in juxtamedullary nephrons, loop of Henle penetrates deep into the medulla before coming back. In cortical nephrons, the descending loop of Henle contains thin segment and ascending loop of Henle contains thick segment, whereas in juxtamedullary nephrons, both descending loop of Henle and ascending loop of Henle contain thin segments. In cortical nephrons, the vascular supply is in the form of peritubular capillary plexus, whereas in juxtamedullary nephron, the vascular supply is in the form of peritubular capillaries as well as vasa recta. Peritubular capillaries around the tubules such as PCT, proximal convoluted tubule, DCT, distal convoluted tubule, and CT, collecting tubule, etc. and the vasa recta along the loop of Henle. In cortical nephrons, the rate of filtration is slow, whereas in juxtamedullary nephrons, the rate of filtration is high and is important in the counter system by which the kidneys concentrate urine, also known as countercurrent mechanism. Coming to blood supply, the renal artery, after entering into the kidney, it divides and redivides to form various efferent renal arterioles. This efferent arteriole enters the Bowman's capsule and forms glomerulus. Now, the arteriole which leaves the glomerulus is known as efferent renal arteriole. It has a narrow lumen than the efferent renal arteriole. The efferent renal arteriole further breaks up or divides into capillaries, and the capillaries around the loop of Henle is known as vasa recta. All the capillaries in turn unite to form the renal vein. This renal vein leaves the kidney through hilum. Vasa recta. The efferent arterioles break up into capillaries around the loop of Henle. We have already discussed the capillaries which are around this loop of Henle is known as vasa recta. Vasa recta is absent or highly reduced in the cortical nephrons. It helps to retain reabsorbed ions and urea in the interstitial fluid of the medulla. Ureter. Ureter is the narrow, distensible tube emerging from the hilum. It is 2.5 centimeters long and 5 millimeters in diameter. So, ureters are the long, narrow, distensible tube which is emerging from the hilum of the kidney. It is basically a metanephric duct which is lined with transitional epithelium. Its main function being muscular, it undergoes peristalsis to pass urine from kidney to the urinary bladder. It comprises mainly of 3 layers: outer fibrous coat of connective tissue, middle layer comprising of smooth muscle, and inner layer which comprises of mucosa or mucous membrane. Urinary bladder. This is the urinary bladder. It is single, median, pyriform bag. So bladder has a shape of pyriform in the pelvic area. This bag has the capacity of 300 to 800 ml. It has 2 parts, upper and lower. Upper body with 2 openings of ureters. So this is the upper part where are the 2 openings of ureter inside the bladder, and the lower trigone leading to urethra and guarded by sphincter. So this is the lower part, and this triangular region is the trigone. The lower part leads into the urethra, and here it is guarded by a sphincter. The wall of the bladder is stretchable, muscular, and lined with stratified transitional epithelium. The ureters and the urinary bladder are lined by transitional epithelium, as this epithelium has a special property of allowing the bladder wall to enlarge. And again, after voiding of urine, the bladder wall comes back to its normal position. Urinary bladder comprises of 2 sphincters. One is the internal sphincter and other is the external sphincter. The internal sphincter occurs around the opening of the urethra, whereas the external sphincter is inferior to the internal sphincter. It is composed of skeletal muscle. Mainly the external sphincter is responsible for the voluntary control of urine. Urethra. The tube emerging from urinary bladder to release urine, having sphincters at both ends, is known as urethra. So we have already seen that from the kidneys, the ureters enter into the bladder. So these are the ureters opening into the bladder. This part is the urethra. This urethra has 2 sphincters at both the ends. One is the internal sphincter and other one is the external sphincter. Internal sphincter occurs around the opening of urethra, whereas external sphincter is inferior to the internal sphincter. It is composed of skeletal muscle. Differences of urethra in men and women. In women, the urethra is a short tube of nearly 4 centimeters in length, opening separately below the clitoris in the vulva. As the urethra is a short tube, more infections ascend and are more prone to urinary tract infections. Whereas in men, urethra is a long tube, approximately 20 centimeters in length, which also serves as reproductive tract it is divided into three parts: upper prostatic part, middle membranous part, and lower penile part. So here is the diagram of urethra in men and women. The urethra of men is divided into three parts. The first part, this is the urethra which is surrounded by the prostate gland. So this part is known as upper prostatic part, and the middle part is the membranous part, and the longest one is the penile part. This entire urethra is of 20 centimeter in length. In men, urethra carries urine during urination and sperms during ejaculation. So, urethra carries both urine and sperms. So, here the urinary system and the reproductive system both are connected in men. Whereas in case of women, the urethra is a short tube of 4 centimeters. In women, the reproductive and urinary system are not connected. They are separate. Tubular reabsorption, also known as selective reabsorption of the total renal plasma flow, which is nearly about 900 liters, the ultrafiltrate constitutes about 1/5 of the total renal plasma flow, which is equal to 180 liters, entering the kidney at the rate of 700 ml per minute. Out of the 180 liters of ultrafiltrate, only 1 to 2 liters is released as urine. This means that most part of the ultrafiltrate is reabsorbed back into the blood, and thus the composition of urine is different from ultrafiltrate. So most of the substances are reabsorbed back into the blood, and this process is known as tubular reabsorption or selective reabsorption. Selective reabsorption involves both passive and active transport of solutes. In different part of tubule, it occurs differently. Selective reabsorption will be discussed in the following parts of the tubule: proximal convoluted tubule, loop of Henle, distal convoluted tubule, and collecting tubule. Coming to proximal convoluted tubule, as we have already seen that PCT is lined by cone-shaped brush border cells. These brush borders can also be called as microvilli. The function of this microvilli is to reabsorb substances back into blood. This is a proximal tubule. This cell is lined with cone-shaped cells which have brush borders, and this is a surrounding vessel. These microvilli help in the absorption of substances into the blood. Coming to the proximal convoluted tubule, 65% of water is reabsorbed back into the blood from the proximal convoluted tubule. 65% of the ions such as Na+, K+, sodium ions, potassium ions are reabsorbed back into the blood by sodium-potassium pumps through sympathors. Some other ions such as chloride ion, bicarbonate ion, and calcium and magnesium ions are also reabsorbed back into the blood from the proximal convoluted tubule. And the essential substances such as glucose, amino acids are reabsorbed completely, that is 100% of glucose and 100% of amino acids are reabsorbed back into the blood. The substances which are secreted into the tubule are: urea is secreted 50% in the tubule and 50% of it is reabsorbed back. Creatinine and ions such as H+ ions and ammonium ions. All these are secreted back into the tubule into the urine. In PCT, that is proximal convoluted tubule, the microvilli absorb substances like 65% of water by osmosis, 65% of sodium ions and potassium ions through sodium-potassium pump, large amount of calcium ions, magnesium ions, and along with chloride ions and bicarbonate ions. These all are reabsorbed back into the blood from the proximal convoluted tubule. 100% glucose and 100% of amino acid are reabsorbed back into the blood. small amount of urea, uric acid and ammonia are also reabsorbed, but the larger amounts are secreted into the tubule. No sulphate and creatinine are reabsorbed. At the end of PCT, that is proximal convoluted tubule, after reabsorbing all these substances and secreting some of the substances, the tubular fluid is still isotonic to blood, that is 300 milliosmoles per liter. Coming to the loop of Henle, this part is the loop of Henle comprising of the descending limb and the ascending limb. Descending limb, and this is the ascending limb. The descending limb is permeable to water. 15% of water is reabsorbed back into the blood from the descending limb of loop of Henle. But this descending loop is impermeable to the solutes and sodium ions, whereas the ascending limb is impermeable to water, but it is permeable to the solutes such as potassium ions, sodium ions, and chloride ions. So descending limb is permeable to water and 15% of water is reabsorbed by osmosis. It is impermeable to sodium ions and other solutes. And the ascending limb is impermeable to water, whereas it is permeable to K plus ions, that is potassium ions, sodium ions, and chloride ions. The filtrate, which is passing down the descending limb, becomes hypertonic as all the water is reabsorbed back into the blood and the water is secreted out. And when flows up, the filtrate flows up the ascending limb, it becomes hypotonic as it is impermeable to water and water is retained inside, and hence it becomes hypotonic. About 80% of water is reabsorbed through obligatory process in proximal convoluted tubule and loop of Henle. 65% of water is reabsorbed back in the proximal convoluted tubule and 15% of water is reabsorbed from the loop of Henle, and thus giving a total sum of 80% of water getting reabsorbed. In this loop of Henle, a characteristic phenomenon mechanism occurs which is known countercurrent phenomenon. This operates in loop of Henle as in the two parallel limbs, that is the ascending limb and the descending limb, the fluid flows in opposite direction and in contrast concentration. Countercurrent phenomenon. This countercurrent phenomenon operates in the loop of Henle of two parallel limbs in which the fluid runs in the opposite direction and in contrast concentration, and along with this vasa recta. As we have seen, till the proximal convoluted tubule, fluid or the ultrafiltrate is isotonic to the blood, which is 300 milliosmoles. So here it is 300 milliosmoles. As it flows down through the descending limb, we know that descending limb is permeable to water. So 65% of water was reabsorbed from the proximal convoluted tubule and 15% of the water is reabsorbed from the descending limb of loop of Henle. Thus, the concentration from 300 milliosmoles changes to 400 after some absorption of water, and then to 600 and then to 800, and thereby it goes on increasing as it descends to 1000, 1200, 1200. So as the filtrate goes down, the water is absorbed and the concentration of fluid inside the tubules increases. Now as the fluid flows up, the concentration which is of 1200 milliosmoles in the medullary part, this is the cortex and this medullary part, it is the highest at the tip of the loop of Henle. So it peaks at the tip of loop of Henle. As it ascends, the concentration of the fluid decreases to 900, 600 and then to 400. This thick part of ascending limb is impermeable to water, but it is permeable to the ions. So NaCl is liberated out, and thus the concentration thereby goes on decreasing as it ascends up, and again it reaches to the osmolarity of 300 milliosmoles, which secretes the sodium chloride, gets reabsorbed into the vasa recta and thus increasing its concentration. So initially the concentration of blood was 300 milliosmoles per liter, which is isotonic to the fluid or ultrafiltrate in the proximal convoluted tubule. But due to the absorption of sodium chloride, the osmolarity increases, reaches the peak at the tip of the loop of Henle. And this water, which is secreted back or which is reabsorbed into the vasa recta, is responsible for lowering the concentration again. And again it reaches to 300 milliosmoles per liter. So in this countercurrent phenomenon, we can see that the ions which are coming out of the ascending limb makes the peritubular fluid, which is the fluid flowing through the vasa recta, of high concentration or hypertonic. This hypertonic fluid draws more water from the descending limb, and this is the countercurrent phenomenon. The ions coming out of ascending limb makes the peritubular fluid hypertonic, which draws more water from the descending limb, and this is the reason for the desert animals to have longer loops of Henle, which is suitable to conserve more water. At the end of loop of Henle, the tubular fluid is hypotonic, as at the end it comes out from the ascending limb and it is impermeable to water. So water is at the end of the loop of Henle, it is in the ascending limb. In the ascending limb, it is permeable to more of the solutes and ions, and thus it becomes hypotonic, which is nearly 100 to 150 milliosmoles per liter. In distal convoluted tubule, which is this part of the urinary tubule, or the nephron, it is mainly concerned with active reabsorption of sodium ions. The sodium ions are reabsorbed into the blood from the distal convoluted tubule. This reabsorption of sodium is mainly influenced by mineralocorticoids, which is aldosterone. This is a hormone which helps in the reabsorption of sodium ions. Reabsorption of chloride ions also takes place due to electrostatic pull. So along with sodium ions, the chloride ions also are reabsorbed into blood. About 10 to 15% of water is also reabsorbed passively to maintain the osmolarity. Along with this ions, some amount of bicarbonate ions are even reabsorbed by the intercalated cells of the distal convoluted tubule. Some amount of urea is also reabsorbed both by the intercalated cells and the principal cells of distal convoluted tubule. And the substances which are secreted into the urine are potassium ions and hydrogen ions. These are secreted into the urine. The potassium ions are secreted by the principal cells and hydrogen ions are secreted by the intercalated cells of distal convoluted tubule. Some amount of calcium is also reabsorbed in the distal convoluted tubule by the action of parathyroid hormone. The collecting tubule is influenced by ADH, which means antidiuretic hormone. This antidiuretic hormone helps to retain more water if there is fall in water level in our body. This is called as facultative water reabsorption. So most of the water gets reabsorbed back into the blood by the collecting tubule. On intake of drugs and alcohol, these substances suppress the antidiuretic hormone secretion from posterior pituitary, and thus the tubular fluid leaving the collecting duct is dilute when ADH levels is low, and it is concentrated when ADH levels is high. So summarizing the selective reabsorption or tubular reabsorption. The glomerular filtration rate in a renal corpuscle is about 105 to 125 milliliters per minute. of the fluid, and that is isotonic to the blood. The filtered substances are water and all solutes present in the blood except proteins. Proteins are large, so they don't get filtered, including ions, glucose, amino acids, creatinine, and uric acid. Coming to proximal convoluted tubule. In proximal convoluted tubule, the reabsorption of substances into blood are water, which is 65% absorbed by a process known as osmosis. Sodium ions are absorbed 65% by sodium-potassium pumps through symporters and antiporters. Potassium ions, which are absorbed 65% by diffusion. Glucose and amino acids are absorbed completely, that is 100%, by the symporters and facilitated diffusion. The chloride ions get absorbed 50% by diffusion. The bicarbonate ions are reabsorbed 89 to 90% by facilitated diffusion. Urea is reabsorbed up to 50% by diffusion. Calcium and magnesium ions are reabsorbed in variable amounts by diffusion into the blood. After the reabsorption of various substances, proximal convoluted tubule secretes H+ ions through antiporters and ammonium ions through antiporters in variable amounts, whereas urea and creatinine are secreted out in small amounts through diffusion. At the end of PCT, the tubular fluid is isotonic to the blood, which is 300 milliosmoles per liter. Coming to the loop of Henle, in loop of Henle, the reabsorption into the blood occurs of water up to 15% by osmosis in descending limb, as the descending limb is to water, and Na+ ions and K+ ions and chloride ions, that is sodium ions, potassium ions and chloride ions, are reabsorbed into the blood in the ascending limb through the symporters up to 20 to 30% and chloride ions up to 35%, as ascending limb is permeable to solutes and impermeable to water. Bicarbonate ions are reabsorbed up to the level of 10 to 20% by facilitated diffusion. Calcium ions and magnesium ions are reabsorbed in variable amounts by diffusion. After reabsorption of all these substances, urea is secreted into urine in variable amounts, recycling from the collecting duct. At the end of loop of Henle, the tubular fluid is hypotonic, which is 100 to 150 milliosmoles per liter. Coming to the distal convoluted tubule. Here, reabsorption of substances into the blood of water up to 10 to 15% by osmosis. Passively, the water is absorbed, and sodium ions and chloride ions are reabsorbed up to the level of 5% through the symporters. And calcium ions are absorbed in variable amounts as they are stimulated by the parathyroid hormone. The distal convoluted tubule has two types of cells. One is principal cell and other is the intercalated cells. So, coming to the reabsorption of substances in principal cells in late distal tubule and collecting duct. Water up to 5 to 9% is reabsorbed into the blood. The water is reabsorbed by the insertion of water channels which are stimulated by ADH, that is antidiuretic hormone. Sodium ions up to the level of 1 to 4% through sodium-potassium pumps are reabsorbed. Urea is reabsorbed in variable amounts. Secretion into urine. Potassium ions are secreted in variable amounts to adjust for dietary intake through leakage channels. Tubular fluid leaving the collecting duct is dilute when ADH is low and concentrated when ADH level is high. Coming to intercalated cells in late distal tubule and collecting duct. In these cells, reabsorb into the blood bicarbonate ions in variable amounts, and it depends on the H+ secretions, hydrogen ion secretion. Urea. Urea is reabsorbed in variable amounts and recycling it to the loop of Henle. Secretion into urine. Hydrogen ions are secreted into urine in variable amounts to maintain acid-base homeostasis through H+ pumps or hydrogen ion pumps.
Hey there! Hope you understood the concept. Here's the solution to the question asked at the beginning. Found this video useful? Hit the like and share icons to enjoy more such videos. LearnPedia's JEE and NEET prep tools contain more than 4,000 videos and over 20,000 solved examples. These can be accessed online through our website or offline through an SD card or a pen drive. To buy now, visit www.learnpedia.in. You can also experience a free demo of our product before buying.